director of the I'm the co-director of the Center for RNA Biomedicine here at Michigan. And I'm very excited to introduce to you today two really excellent members of our center. So we're going to start with Kristen Kutmu, who is an assistant professor in the Department of Chemistry here at Michigan. And just a short bio of her academic road here to Michigan. She started with her undergraduate in chemistry at the University of Colorado in Denver, came to Michigan to do her PhD thesis in chemistry, then did a postdoc in molecular biology and genetics at Johns Hopkins University, and then came back here to Michigan and became an assistant professor in the Department of Chemistry in 2016. Kristen is really an expert on translation and about processes around translation such as elongation, termination. And today she's going to tell us about their work on the consequences of uridine modifications on translation. So any questions you have during the talk, please put it under the Q&A button and we'll field them afterwards. So go ahead, Kristen, share your screen.
Great. Thank you, Matt. Okay.
Looks good.
All right, I'm just getting up the laser pointer if I can. No? Oh.
We see your arrow there though, so it's okay.
Okay, the arrow will work. There we go. Okay, thank you, Matt, so much for the lovely introduction. I'm really excited to be here today. It's always fun to get to talk to an especially RNA crowd. Cells face the daunting challenge of having to maintain the right number of proteins at the right time. And one general strategy that they use to do this is to chemically modify all three major classes of biomolecules in the central dogma after they're made. When I teach biochemistry, and I'm sure in many other biochemistry classes, we tend to spend a lot of time talking about and thinking about the chemical modifications to DNA and protein, things like methylation and phosphorylation. But as I'm sure comes as no surprise to this crowd, it turns out that RNA modifications are also very important. RNAs can be highly modified, and there have been over 150 different varieties of chemical modifications reported in non-coding RNAs. A small subset of these, around 15, have also been reported to be in protein coding messenger RNAs. And the discovery of these modifications has really been made possible by the advent of deep sequencing techniques that have allowed scientists to begin mapping the location of individual modifications of interest transcriptome-wide. So while we've known about the existence of some mRNA modifications like M6A since the 1970s, there's really been an explosion in interest in this type of modification due to the availability of these new deep sequencing tools. The idea that mRNAs can be modified has really garnered a large amount of interest and ignited the imagination of scientists sort of worldwide, because these modifications have the potential to impact every step in the life cycle of an mRNA after it's made, potentially modulating RNA protein interactions, RNA stability, splicing, or even how the ribosome itself views and decodes a particular mRNA transcript. In line with these many potential functions for mRNA modifications, the most wide-studied mRNA modification, N6-methyladenosine, or M6A, has rapidly been linked to a number of different diseases, ranging from cancers to neurological disorders to cardiovascular diseases, among many, many others. Sequencing studies have really laid the foundation for the mRNA modification field, showing us where modifications can be. Currently, we have maps of where roughly 15 different modifications can reside transcriptome-wide. And these maps have given us a beautiful bird's eye view of the modification landscape in cells. And while we know where modifications can be, these large correlative studies, which show us how modification landscape redistributes in response to cellular conditions and different diseases, can't really tell us what individual modifications are doing, even if these correlative studies suggest that the modification might be important for some biological process. So really, the next horizon for the mRNA modification field is going to be moving beyond this mapping bird's eye view, sort of down to street level, to find out what the molecular level consequences of individual modified sites might be. And my lab takes a reductionist approach towards starting to approach this challenge. We use totally purified, reconstituted components of the cellular machinery, and we look at how RNA modifications change interactions between the RNA and these cellular components. So the overall goal of the work in my lab is really to develop a quantitative foundation for understanding RNA modifications and how they can impact RNA, in particular mRNA and tRNA function. We get at this general idea in 4 different ways in my lab. First, we're developing and implementing mass spectrometry tools in order to identify, map, and quantify RNA modifications. We're also interested in trying to understand how the enzymes that incorporate these modifications select both their non-coding and mRNA targets. We also spend a lot of effort trying to understand how the ribosome is impacted by mRNA modifications and also tRNA modifications, because the ribosome is ultimately the enzyme that is working on these 2 very important substrates. And this is actually a much more difficult question than it may sound, because many of the enzymes that incorporate modifications into mRNAs and tRNAs don't do so just into one species. Many of the enzymes, like depicted in this scheme here, will modify not only mRNAs, but other key components of the translation machinery like tRNAs and ribosomal RNAs as well. This makes it very difficult to just knock out a modifying enzyme of interest in a cell and look at a phenotype and be able to ascribe it to any particular modification. And so my lab gets around this problem by using a totally reconstituted in vitro translation system in which we can add or subtract one modification at a time and look at what the consequences of that modification, either to an mRNA or a tRNA, is. Lastly, we've recently become very interested in trying to understand the consequences of modifications that are found in RNA viruses. But the bulk of my talk today is really going to focus on some of my lab's translational work. We've started off by looking very closely at uridine modifications. And the reason that we selected uridine to look at was because of the discovery of this modification, pseudouridine, in mRNAs. Pseudouridine is one of the most abundant modification in RNAs in general, and it, just as one might expect based on that fact, it's quite abundant in mRNAs. It's almost as abundant as M6A, the most abundant mRNA modification. It's often found in the coding regions of mRNAs, meaning that it's a modification the ribosome is likely to see regularly, and it tends to be enriched at RNA protein binding sites, again suggesting that it's really interacting with the cellular machinery. The pseudouridine modifications that are incorporated tend to be at relatively high occupancy, between about 10, and here I have 70, but I think the highest stoichiometry measured is really about 80% occupancy, which I realize is substoichiometric, but it's actually quite good when you keep in mind that other types of chemical modifications to important biomolecules like proteins are also generally substoichiometric, but still can really affect gene regulation and gene expression. Lastly, the incorporation of pseudouridine varies under different stress conditions, and pseudouridine in mRNAs has also been shown to vary during different stages in development in T. gondii, again suggesting that maybe it's doing something interesting. In addition to pseudouridine, we also chose to look at 5-methyluridine and N1-methyl pseudouridine. We looked at 5-methyluridine because we had discovered by mass spec that it's incorporated into mRNAs, both non-enzymatically and by the enzyme TRM2. Our data are supported by 2 additional studies, one mass spectrometry study and one mapping study, also suggesting that M5U can sometimes be found in mRNAs. And then we also looked at N1-methyl pseudouridine, which does not occur in mRNAs naturally, but is being translated in many people worldwide right now as it's incorporated into current mRNA vaccine technologies and many of the emerging mRNA therapeutic technologies. I want to give a really quick plug for basic science here before I move on, because I want to mention that we really started off looking at pseudouridine, and I had to talk my students into looking at M5U and M1 pseudouridine because they didn't think they were going to be that interesting, and they were sort of doing them as side projects. In the meantime, since we've started, COVID happened and M1 methyl pseudouridine became really important, and we discovered M5 methyluridine in mRNA. So it's just a plug for why doing basic science is really important, because at any time, that fundamental question you were asking might end up being biologically relevant. The work that I'm going to be discussing today is really the accumulation of lots of different pieces of projects in lab, all focused around trying to understand the translation of uridine modifications. And it was accomplished by 3 fantastic graduate students, Monica Franco, Jeremy Monroe, and Tyler Smith, as well as my fabulous lab manager, Dr. Dan Eiler. So the first question that we wanted to ask was simply, what happens when the ribosome encounters one of these uridine modifications? In order to ask this, we used a fully purified in vitro translation system in which we have ribosomes formed with an S35 labeled methionine in the A site and a phenylalanine codon that was either unmodified or modified at all 3 positions across the codon in the P site. We then add in all of the downstream factors necessary for translation to happen and just looked to see how fast that phenylalanine gets incorporated by electrophoretic TLC. Our findings demonstrate that when M5U is present, but only at the 3rd position in the codon, it can slow translation. Similarly, pseudouridine also slowed translation by about 2-fold. However, it did so more uniformly across the codon. Follow-up studies indicate that pseudouridine also reduced the rate constant for GTPase activation by EFTU, which is a key step in tRNA selection and accommodation. Our studies suggesting that pseudouridine slows the ribosome are supported by some cellular work that recently came out, where researchers knocked out the enzyme PUS6, which incorporates pseudouridine both into elongator tRNA methionine and into mRNAs. When this enzyme was knocked out, they saw an overall decrease in protein production in the cell, consistent with the idea that the tRNA has been perturbed and is no longer elongating effectively. However, the part that was interesting to me was not that. The part that was interesting to me was that transcripts that were targeted by PUS6 were now had higher protein levels. These mRNAs now had higher protein levels when that pseudouridine was missing, consistent with the idea that pseudouridine is normally slowing translation in some way to reduce protein production. Additionally, we work with Dr. Yuri Polinikov at the University of Illinois at Chicago to solve a crystal structure of the ribosome bound to a phenylalanine tRNA that was placed either on an unmodified or modified phenylalanine codon. These structures revealed that while the base pairing between the tRNA and the mRNA really wasn't very perturbed, the business end of the tRNA, that 3' CCA that brings the amino the amino acid into the ribosome was poised very differently in the peptidyl transfer center on the unmodified and modified mRNA. Normally, that tRNA 3' CCA end is very well ordered and poised and ready to react. This is not what we saw on tRNAs that were bound to our pseudouridinated codon. It's the type of thing crystallographers typically see when there's a problem with tRNA accommodation. And again, further supports our findings that pseudouridine likely slows the ribosome. So we're going to move on now from pseudouridine to M1 pseudouridine, which does not seem to slow the ribosome so much. And if anything, increases the rate constant, though it's by less than twofold. So I would say that's all sort of a wash. So M1 pseudouridine certainly had less of an effect on cognate tRNA addition. We worked with Dr. Aaron Frank here in the biophysics department at the University of Michigan in order to look at some of the interactions between these codons and the tRNA using the structure we collected with Uri for our modeling. And these calculations demonstrated that for when pseudouridine is present at the third position in the codon, there were altered energetic reactions that were not favorable to promoting interactions between the tRNA and mRNA, while we had about 3 kcals of favorable energy when M1 pseudouridine was substituted at that same position. All right, so everything I've told you so far indicates that these modifications can slow the ribosome. And that's interesting. That could be a way for the cell to change protein levels, perhaps, or to cause a pause for co-translational folding. And all of that is good and well. But the thing that I was really interested in when we started off with this was the idea that perhaps modifications in mRNAs might change not only the speed of amino acid addition, but also the fidelity, the accuracy of amino acid addition. And the reason that we thought that pseudouridine might be a really good candidate for this is because pseudouiridine's been studied for a very long time for its amazing ability to make non-canonical base pairs. And because the ribosome relies heavily on base pairing for selecting the correct tRNA, we thought that perhaps by having pseudouridine in a codon, it might be able to change which tRNAs the ribosome was selecting. So in order to ask this question, we again turn to our in vitro assay. In this assay, we have our complexes formed the exact same way as we did in the previous set of studies, only now, instead of providing the ribosome only the correct cognate phenylalanine tRNA, we charged total tRNAs, fed them all to the ribosome, and just looked in an unbiased manner to see what gets made. Do we only make the expected MET-Phe dipeptide, or do we have alternative amino acid selection and make what I like to refer to as a lot of other stuff? Of course, we made a lot of other stuff, or I wouldn't be talking about it. Shown on the left is an electrophoretic TLC with the dipeptide products from our translation reactions. As you can see, we formed quite a bit of the correct MET-Phe dipeptide. However, we also made many substituted peptides. We next went on to see which amino acids were getting added. And we saw that valine, leucine, and isoleucine were also added on codons containing pseudouridine, while serine was not. We see more or less the same pattern of behavior when M1 pseudouridine is present. So shown here are products of 2 TLCs in which the ribosome is translating an unsubstituted and substituted phenylalanine codon. You see we make quite a bit of the correct phenylalanine product and low levels of miscoded product when UUU is present. However, when the first U is substituted for an M1 pseudouridine, we see quite a bit more isoleucine and valine dipeptide being made, suggesting that M1 pseudo-U was promoting the misincorporation of these 2 particular amino acids. We went on to measure the rate constants for misincorporation of isoleucine on pseudouridinated M1 pseudo-U and M5U containing phenylalanine codons. These assays are more or less the same idea as the phenylalanine assays, only now instead of providing the ribosome the correct cognate tRNA, we're only giving it purified isoleucine tRNA. When we do this, we see that the rate constant for isoleucine misincorporation is increased by twofold when M5U is incorporated at the third position in the codon. We see for pseudouridine that isoleucine misincorporation is also increased, but only when pseudouridine is at the first and third positions in the codon. This is not the case when pseudouridine is at the second. When pseudouridine is incorporated between those two U's, all of a sudden the ribosome selects against isoleucine. We see the exact same pattern of behavior with M1 pseudo-U, suggesting that it's something having to do either with this nitrogen or most likely with this nitrogen, because pseudouridine is clearly not using its extra Watson-Crick base pairing phase in order to cause this phenomenon. Just to sort of drive this home, I wanted to show you the curves for M1 pseudouridine, demonstrating that it really depends on which tRNA is being selected and where the modification is within the codon, what effect it is you're going to get. So what I'm showing here now is pseudouridine reacting with 3 near cognate tRNAs that we've each purified individually. When it reacts with serine, you see absolutely no difference between the modified and unmodified codon. When it reacts with leucine, you see that the M1 pseudouridynylated mRNAs cause the leucine to be added a little bit more slowly, but only when it's in the second position. And then with isoleucine, you see all sorts of different effects just simply by moving the modification by 1 or 2 positions, where the isoleucine is incorporated more quickly when M1 pseudo-U is at the first position, and much less quickly, 10-fold less quickly when it's at the second position. So as an in vitro biochemist, it's always very important to verify what it is we're seeing in the test tube with all of our purified components to see if that actually happens inside of a cell. And in order to do this, we work with Dr. Bejoita Roy at New England Biolabs and her team. Dr. Roy and her team in vitro transcribed luciferase mRNAs that contained either only uridine, only pseudouridine, or only M1 pseudouridine. They then transfected cells with these RNAs, HEC293 cells, purified out the full-length products, and subjected those protein products to mass spectrometry. They then ran an unbiased search of each position in the peptide and looked to see which amino acids were getting incorporated. Shown here on the top is the peptide that flew the best, for which they had the best sequencing depth. And suffice to say before I begin, they see basically the same thing we do in vitro, which is that they get much more amino acid substitution on mRNAs that contain M1 pseudo-U and pseudouridine. So in this peptide I have shown here, all of the codons that contain a U that's been substituted are bolded. Those that contain a substitution of a U for a modification where they actually saw an amino acid substitution are shown in red. And as you can see, there are several positions where they did see substitutions, amino acid substitutions, but it's important to note, just like what we saw in vitro, this does not occur at every position. So just to walk you through the comparison of the data, on the unmodified U-containing codons, they saw very low levels of amino acid substitution, as one might expect, with less than 0.05% of the positions being substituted, and they only saw substitutions on valine codons, versus on the peptides that were generated from M1 pseudouridanylated or pseudouridanylated codons, where they saw about 1 to 1.5% of the amino acids being substituted on a much wider variety of codons, up to 18 different codons. So just to summarize what it is we've been discussing, what our data that I've demonstrated, or that I've showed you, demonstrates is that the inclusion of uridine modifications really can change both the speed and accuracy of the ribosome. And it's easy to think about reasons that it might be advantage for the cell to change the speed of the ribosome, right? You can change protein folding, you could change protein expression. However, it's more of a conundrum to me why this might be the case. Also for changing the accuracy of the ribosome. What's the advantage of making the ribosome less accurate in a programmed way where you have hundreds of these modifications all over the place. And so we thought long and hard about this, about what the biological consequences of this might be. And this is sort of where we're at right now. Our idea is that it might actually be advantageous to cells under different conditions to have a little bit of wiggle room, to be able to make a little bit more of a diverse proteome, particularly under stress, without having to go in and change and mutate the genome. We know that miscoding or the misincorporation of amino acids enhances cellular fitness in microbial systems in response to different stresses. We know that pseudouridine in mRNA is increased under different stress and that these levels redistribute and vary with cellular stresses, even in eukaryotes. And we know that there's a negative correlation between yeast fitness and robustness, meaning that if the ribosome is has too high accuracy, it's not so good for yeast in the long term. And that's where I'm going to wrap up today. I just wanted to slightly give a plug and say these are not the only modifications we're working on. We're looking for more members to join our team to look at tRNA and mRNA modifications in different steps in the translation pathway. We've already got some really exciting work going on with the modifications I have highlighted here in red. So there's going to be a lot more of this sort of thing coming, not just the uridine modifications. Thank you so much to my wonderful group, to my funding sources, and all of you for your time.
Thank you, Kristin. It was for a wonderful talk. We have a few questions here. We have time for a few. So one here in the Q&A, the first one is from Dan O'Reilly. Have you looked at non-natural RNA modifications such as those used for RNA therapeutics? These seems to stabilize the RNA against nucleases, and they will be interesting for RNA therapeutic applications.
Yes, so we are definitely, of course, M1 pseudo-U is already one that's being used for mRNA therapeutic applications. And that's one I just talked about here. But we are looking at others as well. We actually have 20 different natural and non-natural modifications in lab, and we're just systematically going through them. If there's one in particular you're interested in, I'd be happy to talk about it offline.
Okay, and Katerina has a question. Is there a consistent pattern in which tRNAs are misreading the modified U containing codons?
So that is a wonderful question, and that is what we are now trying to figure out. So I didn't have time to talk about it, but we've started to do some molecular simulations to help to try to figure that out. A lot of it seems to be based on really physical principles. I've been surprised how well the MD simulations and changes in energy have matched what we've been seeing in our assays, but we don't know what the consistent pattern is yet. I suspect there will be, but we're not there quite yet. That's an excellent question. That's something we're looking into. And I see Wendy was asking, the data indicate that pseudouridine at the +2 position in codons increases fidelity. Yep, that is what we see. At +1, and so at the first and third position, they decrease fidelity. At the second position, they increase it. But I can only say that for isoleucine. So we have another paper that's going to be coming out soon where we look at a broad selection of them. And it really depends on what tRNA it's interacting with.
Okay, we have time for maybe one more question here. Jisoo Lin is asking, are there evidence showing these modifications change RNA structure and change translation stability via RNA structure?
So these modifications certainly can change RNA structure. In the context of what we're looking at right now, I don't think so, because when you think of RNA structure changing it, you often think of something downstream of the RNA. And here we've formed complexes on top of those pseudouridines. What I can tell you is that our molecular dynamics simulations, together with Professor Aaron Frank here at UMich, really suggest that it's not the structure that's changed so much as the entropy of the mRNAs that seems to be having a large effect.
Okay, we have a few more here, but I think we'll do them offline. I had just one quick question. So are all these modifications happening post-transcription, or are
Yes.
some of them happening to the pool of ribonucleotides?
So that is an interesting question. That's something people are looking into, particularly for methylations. For pseudouridine, though, no. That's
It's
all post-transcription.
all post. Okay. Okay, then. So thank you so much, Kristen, for that. And we're now going to switch over. Our second speaker today is Peter Todd. And he's a professor in the Department of Neurology. Just a quick run through of how he got to Michigan. He did his undergraduate work in Southern California at UC California at San Diego. He did then his MD and PhD work at University of Wisconsin in Madison, and trained as an intern and in residency at the University of Pennsylvania in Department of Neurology. And then we were fortunate to recruit Peter to University of Michigan, where he started as an assistant professor in 2010, and he has risen to full professor as of last year. And Peter's expertise is really the role of repetitive sequences in neuronal functions and their expansion that causes various diseases. And today, Peter's going to tell us about repeat-associated non-AUG initiated RAN translation in neurodegenerative disease. So go ahead, Peter.
Great, thanks, Mats. Do you see my slides okay? Everything look all right?
Yes, it looks like you're in a warm spring day in Ann Arbor.
Oh, yes, it's
not
important.
correct.
Yeah, maybe not. Myths are important.
Yeah.
So first, I just want to take a quick moment to shout out to all of the AAAS fellows that were elected today, and note that we had 20 new fellows elected here at the University of Michigan, and that actually includes 4 fellows that are part of the Center for RNA Biomedicine. I just want to highlight the one in the bottom corner in particular, our host today, Mats Lundman. So congratulations, Mats, and to all the AAAS fellows elected this year, both at Michigan and across the country.
Thanks, Peter.
Yep. So my lab is interested in, I'm just going to minimize the picture of me. My lab is interested in repetitive elements in the genome and how they contribute to human disease. And I'm a neurologist, so I get to see patients with these diseases in clinic and then trying to do research on them to develop therapies back in the lab. Repeat expansions can happen at many different sites throughout the genome, both in coding regions and outside of coding regions, and both within and outside of genes. Most of the repeats that we know that are associated with disease occur within gene regions. Repeats can happen in coding regions themselves, such as the trinucleotide CAG repeats in Huntington's disease and many of the spinocerebellar ataxias. And there's a large number of repetitive elements that range in size between 3 repeats out to 10 repetitive elements that can occur throughout genes in both 5' UTR intronic and 3' UTR sequences. The field of repeat expansion diseases is itself actually expanding. So we're seeing the identification of new repetitive elements that are associated with human diseases arising on a regular basis through improvements in sequencing technology. And these often end up being common or relatively common causes of neurologic conditions. The mechanisms by which repeat elements can cause human disease has been a focus of study for the last 30 years, since the Fragile X locus was described as a repetitive element that's associated with disease. And repeats can occur and cause problems through a variety of means. So at the DNA level, repetitive elements can become methylated, and that can lead to transcriptional silencing of the locus, and can also cause problems with the transcriptional event through R-loop formation and triggering of DNA damage responses. As RNA, the repetitive elements can form secondary structures, including G quadruplexes and hairpin structures that can bind to specific RNA binding proteins and then sequester them into RNA foci structures in the nuclei or the cytoplasm and preclude them from doing their normal functions. When repeats are located within coding regions, as occurs in Huntington's disease, the expansions of those repeats lead to long stretches of typically homopolymeric proteins, often polyglutamines, and those elicit a toxic gain of function where they change the negative functions of the proteins in which they reside to take on new forms of toxicity, while also causing aberrancy in protein quality control mechanisms. My own lab has focused a lot of energy in the last decade on the study of an unusual mechanism by which repeats can cause disease, known as RAN translation, or repeat-associated non-AUG translation. And that'll be the focus of much of my talk for the rest of the day. So what is RAN translation? So RAN translation was first described by Tao Zhu and Laura Ranum's group at the University of Florida when they were studying a CAG repeat in the gene spinocerebellar ataxia type 8. What they did was this gene had an unusual structure where the ATG was immediately before this long stretch of repeats. And if they mutated that ATG codon to an AAG, they saw that they still got production of protein from this locus. They then went on to show that actually proteins could be produced in all 3 potential reading frames from that CAG repeat in the absence of an AAG start codon through a mechanism that at the time was mysterious. And we're able to show that products from these appeared to accumulate in patients with spinocerebellar ataxia type 8. Our own group had was at the time, I was a fellow in Hank Paulson's lab, was doing studies on a related disease known as Fragile X tremor ataxia syndrome. And in this condition, the repeat actually resides within the 5' UTR of FMR1 and is a CGG or GGC repeat. And so a group, Peng Jin and Steve Warren, had made a Drosophila model of this where they'd placed that 5' UTR in front of GFP and expressed it in flies. This led to a degenerative eye phenotype that you can see here on the right. When I got a hold of these flies, I began doing some experiments. I was surprised to see that actually in the Drosophila eye, the GFP was actually forming inclusions or aggregates in the cells in which it was being expressed, which was unusual and different from what I would see with GFP expressed in the context of other proteins. These inclusions were present in both the nucleus and the cytoplasm, and they co-localized with markers of protein quality control failure, including ubiquitin and HSP70. And I should mention in FACS-TAS that there actually are, the pathologic hallmark in this condition is actually ubiquitinated and HSP70 positive inclusions in patient brains, in the neurons. So to try and understand what was going on at that time, we ground up the flies and ran them on a Western blot and discovered that in the flies that had this expanded repeat, there was this higher molecular weight band or species that was present that was not supposed to be there. And through subsequent experiments, we're able to demonstrate that this was actually due to aberrant translation of the repeating element in a fusion with GFP to create a polyglycine product that aggregated both in the flies and in humans. After those initial discoveries of repeats at the CGG locus and the SCA8 locus, multiple groups have now identified that RAN translation can occur in many different contexts, both within coding regions, as in diseases like Huntington's disease, in intronic sequences, as occurs in the hexanucleotide repeat associated with ALS, and even in some sequences within 3' UTRs. I'm going to talk to you today about 2 of these repeats, a CGG repeat that causes Fragile X tremor ataxia syndrome, that I just mentioned, as well as the hexanucleotide G4C2 repeat that's present in the first intron of C9orf72 and is the most common genetic cause of ALS. So to give you some context of how this might work, I'm just going to go over a brief review of how regular translation works. So as you guys all know, RNAs have a 5' methyl G cap, which binds to the EIF4F complex and recruits the pre-initiation complex, including the 40S ribosome, as well as the ternary complex that features the tRNA methionine. This complex then moves along the RNA in a process that involves helicases unwinding and allowing the RNA to, the complex to move in a 5' to 3' direction until it finds an AUG codon in an appropriate sequence context. That then leads to selection and changes in the structure of a number of proteins, including EIF1 and 5, and kicking out of some factors and recruitment of the large subunit of the ribosome, translation until a stop codon is reached, and then release of the protein for its function. Our working model for how RAN translation works actually is sort of 2 working models. So the first is a process that actually models very much what we see in regular translation. So still requiring that cap, binding with the EIF4F complex, recruitment of the 43S ribosome, and a need for scanning along that RNA. In this context, these repetitive elements form a strong secondary structure that actually impedes that translocation or scanning process of the pre-initiation complex. And that leads to enrichment or use of near cognate codons for initiation. And this is such as basically a failure in start codon fidelity elicited by a strong secondary structure in the RNA. There's then recruitment of the rest of the ribosome and translation through the repetitive element to make homopolymeric proteins in the case of a trinucleotide repeat like CGGs, or dipeptide repeats in the case of a hexanucleotide repeat like G4C2 sequences. At the same time, we know that there is some initiation that occurs within the repeat itself at codons that look nothing like AUG, and those events are less efficient but still present. In addition, elimination of that cap does not completely preclude initiation by RAN translation. And so it's likely that an overlap of these two mechanisms are at play in any given transcript, depending on its sequence context. So there's been data now from a number of groups that have shown that the products of RAN translation can accumulate in human brain tissue, and that expression of those products exogenous to the repeating elements is sufficient to elicit neurotoxicity. So if RAN translation products actually contribute to human disease, and if we could identify factors that are selectively required for RAN translation, we reason that those factors would serve as potential targets for therapeutic intervention. And so this set of ideas has driven a lot of our work over the last decade. To do this, we've taken a couple different approaches. So first is we developed a simple reporter assay for measuring RAN translation, where we took nynoluciferase and mutated the AUG codon at its start to a GGG that's not used readily for translation, and then placed that 5' UTR of FMR1 or placed other repeat elements above this sequence, and then introduced small 1 or 2 nucleotide frame shifts between that 5' UTR sequence and the nynoluciferase sequence to be able to detect products in different reading frames. And so you can see here that we create a higher molecular weight species that's initiating above the repeat and increasing in size at increasing repeat sizes. This event is relatively inefficient, so the total efficiency of translation by RAN translation is about 1/50th of our AUG nynoluciferase sequence, but is still 100 to 200-fold over our negative control construct. Translation in different reading frames shows different efficiencies. We can also take this assay and do both in vitro assays, cell-based assays, and studies in neurons that allow us to sort of look at different variations. So we coupled that with this Drosophila model, which allowed us to utilize factors we predicted might play roles in RAN translation, and then altered their expression selectively using available either RNA lines or overexpression systems. So I'm going to summarize a lot of published work in a few slides here. So the first set of findings suggested that a number of different RNA helicases that play roles in unwinding the repeat actually significantly impact the efficiency of RAN translation. So knockdown of EIF4B or 4H or DDX3 led to a decrease in RAN translation from CGG repeats. At the same time, if we modified factors that have selective roles in start codon fidelity, EIF1 and EIF5, we saw reciprocal effects. So EIF1 enhances codon fidelity, its overexpression suppresses RAN translation from these reporter assays, and EIF5 suppresses codon fidelity and enhances RAN translation. We've recently followed up on those initial findings to show that another related protein, EIF5 mimic protein, which might be more targetable therapeutically, also has similar effects on codon fidelity and on RAN translation at CGG repeats. This is work by Becky Gleinberg in association with Katsura Asano. At the same time, our group and others identified that responses to the integrated stress response, which normally suppresses global translation, had a paradoxical effect of increasing RAN translation. So normally the tRNA complex is brought into that pre-initiation complex. In the context of integrated stress response activation, the EIF2 alpha is phosphorylated, which slows the rates of recycling of this tRNA complex and decreases initiation rates as a way to sort of globally slow translation. However, RAN translation with both initiation within the repeat and at near cognate codons above the repeat are markedly enhanced in the context of EI2 alpha's phosphorylation, suggesting that perhaps a freeing up of translational factors allow for this relatively inefficient event to occur more frequently. To try and understand how that process might work, we and others have been exploring the possibility that alternative ternary complexes or factors that can bring an initiator tRNA to the initiation site might be used in the context of this stress event. And so a number of different proteins, including EIF2A, EIF2D, and the Demer MCTS1 complex are ones we've looked at. And in particular, we found that the Demer MCTS1 complex seems to play an important role in this process. So if we knock down Dener, we see that we get a marked decrease here in RAN translation from a hexanucleotide repeat associated with ALS. That is dependent on that start codon. So if we place an AUG codon above that repeat in a good Kozak context, we don't see any effect of this Dener knockdown. We can also see a very similar set of results if we knock down the cognate protein MCTS1. Knockdown of any of these alternative ternary complexes actually modestly diminishes production of RAN products and in fly models, and modestly improves phenotypes. Although in none of these cases did we see a marked improvement in survival with these corrections, either through assays in eye phenotypes or survival assays. So a second set of directions that we've gone to try and understand how this RAN translation event occurs is actually looked at the RNA molecule itself. And this sort of raised an interesting question for us related to, especially to this hexanucleotide repeat in ALS. So there was a question as to what structure this actually forms in cells. So we're pretty confident that this can form a G quadruplex structure quite avidly when you put it in vitro, but there's been a question about how often that happens in a cellular context. To try and look at that, Indranil Malik in our lab, in collaboration with Sylvie Ruskin's group, used a DMS map-seq method to selectively modify bases and then look for base pair changes that would occur when there was these were reverse transcribed using a TGRT. So the base idea here is that with DMS actually leads to modification of both unpaired A's and C's, and then we would see a change in those bases if those modifications occur. In a sort of base sequence with the surrounding intronic sequence, but absent any repeats, we could see readily modifications at many sites. However, when we try to do this at very large repeat sizes, with large G4C2 repeats, we appear to not get any modifications, but we got very little signal, probably due to differences and difficulties in both sequencing and getting the reverse transcriptase through this repetitive element. To try and overcome that, we used a couple of shorter constructs, one of which had 8 G4C2 repeats, and another of which had 20 repeats with a single A modification. So for the 8 repeat one, we were able to get decent coverage over this region, and actually able to sort of figure out the structure, which was predicted to be predominantly a single hairpin structure rather than a quadruplex forming in this region. In this 20 repeat construct, a single A nucleotide was sort of spuriously introduced about 2/3 of the way through this repeating element, and that turned out to be very helpful because when we actually did our sequencing here, you can see this single very large red peak, which is that modified A. And again, this A modification suggests that's highly unpaired and that we're sequencing through this, and is predictive of this being a hairpin structure, not a quadruplex structure in this context, as we would expect the surrounding C's to be unpaired in a quadruplex structure. So if this is actually forming different structures in cells than we might have predicted in vitro, what does this tell us about the effects on its translation? So to look at this, what Indranil did was he took the repeat RNA and he folded it either in a sodium or potassium buffer to favor formation of hairpins or quadruplexes. And by doing so, he actually led to the, he could then detect what effect this folding had. So when we folded in a potassium solution, he markedly reduced the efficiency of translation that occurred, whereas a sodium, compared to a folding in a sodium solution or no salt buffer. This was true not only in that GA reading frame, but across multiple reading frames with this hexanucleotide repeat sequence. And he could sort of redemonstrate that this was actually due to the folding by taking those RNAs he'd folded in a sodium or potassium solution and then heating them up and showing that those quadruplex structures would actually resolve and allow for translation after denaturing of those structures. To try and see if this also held up in cells, he pre-folded RNAs that were in vitro transcribed and then transfected those directly into cells and saw a more modest effect of those pre-folded constructs that have a G quadruplex form versus a sodium form, suggesting that there is perhaps less effect in an in vivo situation, although there's some caveats to this piece of data. So if a G quadruplex structure impedes RAN translation from G4C2 repeats, then we would expect that loss of RNA helicases, which resolve G4C2 repeats, would be expected to suppress RAN translation. And so to try and get at that question, a talented graduate student in the lab, Lulu Cheng, explored a potential role of a protein known as GHX36 or the G4R1 resolvase. And this protein is a G quadruplex specific resolvase that affects both DNA and RNA structures. When knocking down this in cells, what you could see was a significant reduction in the efficiency of RAN translation when we knocked down DHX36, which would be predicted to increase the number of quadruplex structures that would form from this repetitive element. And that was true across multiple reading frames and also true for CTG repeats, which are capable of forming a quadruplex in vitro. This did not significantly affect the expression, at least of our reporter assays, of the RNA molecules being expressed in these contexts, suggesting this was predominantly a post-transcriptional effect. And this was true also in a repeat-dependent fashion. So if we used constructs that had shorter repeats or elongating constructs, we saw that only larger repeats that would be capable of forming quadruplex would have a significant effect of this DHX36 knockdown. We also saw the opposite effect when we overexpressed DHX36. So here by supplying an overexpression of the wild type protein, we saw a marked increase in RAN translation efficiencies across reading frames of this hexanucleotide repeat sequence. But a mutant form of DHX36 that lacks the helicase activity did not have that same effect. Interestingly, the loss of DHX36 also prevented that stress-dependent upregulation that we saw of RAN translation. So we could see a significant increase in RAN translation rates from our reporter assays of both the G4C2 and CTG repeats in control cells, but in cells that had a knockdown of DHX36, we lost that upregulation that occurred in this context. At the same time, Indranil was interested in this idea about how these RNAs were folding in cells and how that might influence their behavior. And he was particularly curious about this idea that RNAs and their interacting partners in cells are likely very different because the ways RNAs are produced in those contexts versus in vitro contexts will lead to very different potential interactions in different compartments and in different cellular contexts and responses to different cellular situations. So to look at this, he developed a series of reporters using a CTG repeat in the 5' UTR with a PP7 tags below the repeat, allowing him to capture the RNA, and then grew cells expressing either the CTG repeat RNA or an RNA lacking that CTG repeat, and then captured those RNAs as associated proteins in different contexts, both in nuclear and cellular or cytoplasmic fractions, and with or without the introduction of stress. What he found was that actually meant while some of the proteins we'd identified through in vitro interactions with CTG repeats were same, there was new sets of proteins that had not been identified through in vitro assays only. And in particular, the SRSF family of proteins were highly enriched as CTG repeat interactors. And those interactions were enhanced even further in the context of cellular stress. He then utilized our Drosophila model and looked at what the effects of knocking down SRSF1 or SRSF2 proteins were on the fly phenotype, and saw a significant rescue of the rough eye phenotype and a survival effects in CTG repeat expressing flies. And if he overexpressed SRSF1, that led to a significant enhancement of that rough eye phenotype, suggesting that this might be a modifier of toxicity. We saw similar effects when he looked at modifying this protein in the context of the hexanucleotide repeat that causes ALS. It turns out SRSF proteins are very important, so their elimination is not really a good therapeutic target, but there was some drugs in development that targeted a protein kinase which modifies the behavior of SRSF. And so this SRPK1 protein actually phosphorylates with SRSF and modifies its distribution into the nucleus, and therefore its potential roles in transporting RNAs. So he reasoned that if we inhibited this enzyme, we might actually influence repeat RNA trafficking, and by doing so, influence RAN translation. So first, in Drosophila, he was able to show that knocking down these proteins was sufficient to rescue those rough eye phenotypes we'd shown earlier, similar to what we'd seen with SRSF protein modifications. And he could see that actually using these drugs in human cells with reporters showed a selective inhibition of RAN translation at both CGG repeats and not shown here at hexanucleotide repeats. To try and understand how that worked, he did a couple of different assays. So first, he could show that this SRPK1 inhibitors did lead to a change in the distribution of SRSF1, and that influenced the distribution of the repeat RNAs such that they were more nuclear in the context of these inhibitors and less cytoplasmic, meaning they didn't have a chance to access the translational machinery for making RAN translation products. Similarly, or somewhat differently, he could also see that in vitro assays and in RNA transfected cells, that these drugs could actually inhibit RAN translation directly, suggesting that not only was it inhibiting its transport, but its actual translation. And lastly, in collaboration with Semi Biomarta's group, we utilize an automated microscopy group in methodology to track the survival of neurons over time. And what we saw, and which we'd seen previously, is that expression of the CGG repeats, here the y-axis is toxicity, expression of the CGG repeats, which takes this dotted line up to the, or the green line up to the dark solid line, leads to a significant enhancement of toxicity. But the treatment with either of two different SRPK inhibitors basically brought survival back down to our GFP expressing control cells. So to summarize what I've told you today, GC-rich repeats are capable of eliciting translation in the absence of an AUG codon. And there's a number of different things that modify this process, including things that can modify the fidelity of start codon selection, RNA helicases that can affect the structure of the repetitive elements itself and the surrounding sequences, alternative ternary complex factors that might be able to replace EIF2 in context of stress or even under basal conditions and have lower fidelity. And so the combinatorial effects of these are potential targets for development as we begin looking at ways to block RAN translation and hopefully translate to therapeutics. With that, I want to thank the people from my group who did the work. I've highlighted most of these people on the way through. I also want to highlight our collaborators, Sylvie Ruskin at Harvard, who helped with all of the structural data, and Phil Smaldino at Ball State, and Semi Biomarta's group, who's helped with all of the neuronal work. And with that, I'd be happy to take any questions.
Well, thank you, Peter. That was excellent. There are a few questions in the Q&A here. The first one is from Dan. He asks, is there a correlation between I think the output of translation and the number of repeats?
Yes, there is. So we do see that as we increase the repeat size, so it's somewhat different, obviously. So when we get initiation at the near cognate codons that are just before the repeats, there is actually some initiation at those even in the absence of any repeat, as we've known from work from Nick Ngole and other groups across for a long time. But the efficiency of that translation goes up as the repeat size increases, so that we do see an increase of effect, especially at the CGG repeats.
Okay, Bojan has a question. In full mutation Fragile X patients, the FMR1 messenger RNA is generally not expressed. Do you observe a non-AUG translation mostly in pre-mutation patients?
That's correct. So there are Fragile X syndrome, and I didn't go through this because of time, is a developmental disease due to transcriptional silencing of the FMR1 locus. And so in that case, you lose the Fragile X protein FMRP, and that seems to be the cause of disease. There's a separate allelic disease where you have a repeat that's in an intermediate range or a premutation size. And in that case, it's still transcriptionally activated, and that causes this age-related neurodegenerative disease, FAXTAS. And in that repeat, which is expressed as transcribed, is actually undergoes RAN translation associated with the disease. We do not see inclusions in the brains of Fragile X syndrome patients. Or I should say, we usually don't. There are rare inclusions reported, but that may be due to mosaicism in the level of methylation that occurs in some patients.
Okay, Niels said it was a great talk. Are there any cellular stress conditions where potassium levels can get so high that the CGG repeats from quadruplexes overpowering the helicases?
Yeah, we haven't looked at that, so I don't have a good answer to that. I do think that in cells there is probably a wide variety of reasons why we don't see as many quadruplexes form. And work by Junji Guo when he was in David Bartel's group really suggests that that in vivo quadruplex versus in vitro quadruplex formation rates are probably different. So I would suspect that it would be less the potassium level and more that there might be the tendency to form a quadruplex will be directly influenced by the presence of factors which would allow or augment the quadruplex formation.
All right, let's jump to Annalisa has a question. Do you know if the RNA is modified at the expansion?
That's a great question. So we don't know. We've looked a little bit at M6A signaling, but have not been able to see direct modifications. There are in both the repeats I just showed you, obviously they're a G4C2 repeat, so we would expect that a methylation would have to be on the cytosines. And we haven't done anything with with those in particular. There are AGG interruptions in the CG repeat of FAXTAS that could potentially be targets for M6A modification, but there's no data to suggest that's occurring on a regular basis.
Okay, great. Last two questions here. One is from Peng. How does the 40S identify the first start codon to start translation?
That's a good question. So, we know that obviously there's been a lot of work by many groups looking at codon fidelity for years under normal circumstances, dating back to work by Marian Kozak in the 80s and other groups. And so we know that that codon fidelity choice is influenced by a number of different factors. And Alan Hindenbusch has done a lot of work in yeast on these questions related to what can influence those base choices. So EIF1 and EIF5 I talked about. There's also some other initiation factors that can directly influence that process. So the selection, though, does have some surround sequence impact, and we do think that's an important component of this. How could initiation happen at these non-cognate codons? The way we envision it is that basically, the sampling, any given selection event is going to be influenced by the rate of movement of this ribosome. So if a ribosome is stalled or the pre-initiation complex is stalled, that will favor initiation at codons that are not favorable. And at some point, a complete stalling might actually allow for initiation to occur for a codon that doesn't have any sensical reason to be initiate initially. So that's our current working model, but we haven't been able to fully prove that, especially at non-cognate codons yet. So we still have some work to do. I will say that if we modify EIF1 expression, if we increase it, we actually do decrease both translation outside of the repeat at those near cognate codons and in the repeat at non-cognate codons.
Okay, last one. I promised one more. So Jody has a question. Have you looked at the DMS-seq after DHX36 knockdown to see if you see a signature arising from a G quadruplex containing population of RNAs?
Yeah, that's a great question. So we're still trying to really optimize our ability to detect the long repeat sequences. And so that's been sort of a rate limiting step right now on those experiments. But we're hoping to use some newer techniques to do that, that will allow us to actually get reads all the way through the repeat and we'll be able to tease that out, I think, more accurately. But I think you're right. We would expect that to occur, but we don't have direct experimental evidence that it does occur. So I think there's still work to be done in that area. And there's a number of groups that are looking at DHX36 in the context of that hexanucleotide repeat across the country. So I suspect we'll have an answer not too long from now.
All right, great. So I would like to thank everybody attending. We had over 200 at some point, and thank Kristen and Peter for 2 excellent talks. And we'll be back in 2 weeks, I believe. Thank you, and bye-bye from Michigan.